Okay, class. Well, welcome to Snow Day. Hopefully, the video is okay here. This is about the best I can do with my laptop. And most of the time, you won't be seeing me, you'll be seeing slides up on the screen. But before we start, a couple groundkeeping things for over break. First of all, you have my initial email. So today is lecture 12. Please watch this lecture. As well, there's going to be attached worksheet working with Punnett squares. Please do that. And then in terms of your tests, if you did really well, I'll probably just send you your grade over break, and then you can actually have your physical test back. But if you had some problems, I will actually scan your copy of your test and send it to you. Because as I mentioned in the email, I do want people who had some struggles to have the opportunity to raise themselves some. So, as I said in the email, you will have the opportunity to retake one or both tests if you did poorly. I'm going to say that's below what I consider a 79%, and then the two would be averaged. Now, you can look at your old test up until 4 days before you take the new test. I just don't, want you to sort of memorize everything and remember, I really want you to learn the material. The other thing that I will do is send out a review packet it'll be very high level and not have a lot of detail, but it will cover all chapters up to, I believe, chapter 10. And then you can fill that out if you want. If you need extra points, I said I'll give some points if you do it and hand it in. I will apply that to where it helps you the most, whether that's to a take-home quiz or whether that's to a test as well. Then the third thing, when we come back, we will start in chapter 13, but there will be time for us to go over the Punnett square worksheet, as well as any questions you might have from this material. And then also when we come back, we're going to be watching a video, part of the time in the first two classes, I believe, about the life of Gregor Mendel, who we see today in this chapter. Before we start the actual lecture, I read a article in the news last week that was really interesting. It impacts a lot of the topics we've covered in class. So what happened was, and I believe, if I'm remembering correctly, it was a police officer who, sometimes when people have certain forms of cancer, the best possible treatment is to go in and destroy all their existing bone marrow, and then they get a transplant actually from a live donor of some bone marrow. And what that does is your bone marrow is the part of your body that's responsible for creating new blood cells as well as white blood cells. So it's in control of the immune system. So when there are cancers that affect the immune system, or it's believed that the cancer is reoccurring because you have a faulty immune system, the idea is you kill your entire bone marrow, replace it with someone else's, and then it in effect gives you a new immune system, and hopefully you don't get the cancer again. Well, what was interesting about this is one would expect that since the bone marrow is responsible for making new blood cells, white blood cells, that you would find that if you did genetic testing once you had a bone marrow transplant and all your original bone marrow was killed and you have the donor's marrow, that of course your blood cells would no longer have your DNA, but the DNA of the person that donated the bone marrow. But what was unexpected, and since this person was a police officer, he was told to go ahead and get genetic testing because it is a forensic issue. If you bleed on a blood scene and your DNA has been changed, so he was encouraged to get genetic testing, but the surprise was it wasn't only his blood cells that had changed DNA. They found that a lot of his cheek cells, and remember the cheek cells and your nasal cells and your esophageal cells and your intestine cells, those all reproduce really quickly. They have a short cell cycle, as we've said a while ago. A lot of these cells now no longer had the original man's DNA. They had adopted the donor DNA, which is really surprising. We don't know the mechanism for why that happened. Now, of course, other cells in your body, like nerve cells, some of your brain cells you have for life. They never divide. And skin cells, my outer skin, of course, is dead, but it takes 7 years to change your, new get new skin to cycle all the way through because you have several layers of skin. So, some of these slower cells haven't changed. And of course, probably not all of the cheek cells have changed, but some have changed to the donor DNA. And so that was interesting. But the other very interesting thing was, and we talked about meiosis last week, about the process of making gametes, the sperm cells or the egg cells, to reproduce. This man's sperm was no longer his own. It actually adopted the DNA of the donor. So that would mean that if he had children now, they wouldn't biologically be his. They'd biologically be that of the donor. And so, again, that is really interesting. I think people don't know all the implications of this or actually how all this has happened to this man. But, it does, if someone's going to have a bone marrow transplant and wants to be a father, they're going to probably want to take some steps to either store sperm or whatever to have their own genetic children. Now, it wouldn't affect women, at least in terms of egg cells, because remember, the egg cells begin to form when the woman is in her mother's womb. So, but men will, once they've hit puberty, continue to produce sperm. So that was very interesting and unexpected science news. So last week we looked at meiosis primarily, and as you remember, eukaryotes, and especially humans who are eukaryotes, we have two copies of every chromosome. One is from your mother, the maternal copy, and one is from your father, the paternal copy. And so humans have 46 chromosomes, and they are in pairs of 2. And so when we get ready to reproduce, we pare that down to what's known as the haploid number, where there's only one copy of each chromosome. Now, as we saw, when that happens, it's a random sort of splitting of for each chromosomal pair, whether the maternal or the paternal chromosome is passed on. So we have 23 pairs, and there's 2 choices for each pair. So in terms of variability in types of either sperm or egg cells a human being can create, that would be 2 to the 23rd power, which would be roughly 7,600,000. And so there's a lot of variability just from sorting out the possible combinations of one chromosome out of 2 in the pair, and when you have several pairs. And then in addition, we saw that in meiosis, sometimes we had something called crossing over, where when the chromosomes are bundled together during the meiosis process, and you have the pair of the paternal and the pair of the maternal for the same gene, and they're bundled, and the process of that was called synapse. They're packed tightly together. Sometimes the maternal pair and the paternal pair can swap genes. Now remember, a chromosome has several genes on it. So basically what we end up with is when that happens, a new gene that's sequenced differently. It's got, if it's the paternal gene, it's got most the paternal gene, and then uh, maybe a gene from the mother inserted. And usually that's a good thing because, as we remember, we have um, of our 46 chromosomes, 22 of them are autosomal pairs, meaning that each chromosome does exactly the same thing. It might have different traits, but it's responsible for exactly the same thing. Um, the problem occurred, as we saw, when you have an X and a Y chromosome, because they're not autosomal, they're different chromosomes. And so sometimes if you have a portion of the Y chromosome cross over to the X chromosome, it can result in things that are not ideal, as we saw. Um, Now, this week we are going to step back and not look at the microscopic scale of what's happening, but look at how this, genetically works itself out. And we began to talk about that a little bit at the end of class last week, but this week we will be looking at genetics. Um, so, as you remember, we saw the process several weeks ago that we had our DNA, and the DNA was transcribed into messenger RNA, and that was then translated into proteins, and those proteins did things, and they gave you your characteristics. And when that was happening, we said that gene was expressed. And remember, we talked about chromosomes, and chromosomes have several genes on them. It's a sequence of genes. And each individual gene codes specifically for one polypeptide. And then last week, as I mentioned, talked about meiosis and taking these genes, taking them from the diploid number down to a haploid number, and creating the gametes, the sperm or the egg cells, which then combined. And when the egg cell was fertilized, and we called the joining of the genetic material fusion, we had an entirely new genetic code for an individual, and that new fused cell was called a zygote. And the zygote then goes through mitosis, normal cellular division, and grows and grows and grows. You have a new life form, a new human being in the case of humans. So meiosis divides the genetic code you have, and the two parents pass that genetic code down. And in that genetic code are certain traits, and the passing on of that trait or those traits is called heredity. Now, as we've said, a gene is a specific section of the DNA responsible for making the protein, and what exactly those proteins are determine all kinds of characteristics you have. And a gene is expressed when it is going through transcription and translation and making that protein. Genes aren't always expressed, as we said, for various reasons. Sometimes a cell has a certain function where the gene is expressed. So in your pancreas, which is responsible for making insulin, those genes are expressed, but they wouldn't be expressed in your skin cell because your skin cell is not responsible for making insulin. Sometimes a gene is expressed when it is needed. An example is if you break a bone, there's a mechanism in place where then a gene is expressed that makes a protein that tells the bones it's time to heal. Normally, when you don't have broken bones, they don't make that protein because they don't need to heal themselves. So sometimes it's a condition. Other times it might be age. For example, hair color. You reach a certain age and the gene to code whatever color your hair has stops being expressed. And the result is you eventually end up with mostly gray or even white hair. Um, so then that brings us to, as we talk about genes, the entire sequence of an organism and the entire sequence of its DNA is known as its genome. So that there's that term genome. Um, it's the entire set of the organism's genes. It's a very large number of genes, even for simple organisms. Now, your textbook says that the human genome is around 30,000. And this probably reflects older data. It's kind of interesting if you were to look back in 1960 when DNA was in the 50s was when Watson and Crick determined the structure of DNA, and we're beginning to learn about DNA. It was estimated that there were 6,700,000 genes. In 1990, that was when we actually took the genetic testing technology that we had and began to test for genes and map the genome. That was the beginning of the Human Genome Project. That was in 1990, and at the time in 1990, that estimate of 6,700,000 was revised down to 100,000. And over time, and we've gotten sort of now, and it took a few decades to map the genome. We've got it mapped, but there's still debates over what constitutes a gene and what doesn't. There's debates over methodology and things of that sort. But the count gradually went down to 30,000, then it went down to 20,000. Now it's up to 21,306. But there are going to likely be changes because there's still debate over this number. But remember, as we said, not every one of these 21,306 genes, if that's the right number, will be expressed at all times by all cells in your body. Cells will only express genes related to the proteins that are related to the functions they need. And remember well that we have mentioned our genome, and I've said this several times, is diploid, possessing both a maternal and paternal copy of every chromosome, and subsequently every gene. When it's time for sexual reproduction, as we mentioned, meiosis will randomly mix a new combination of maternal and paternal chromosomes to pass on in a haploid gamete cell, whether that's the sperm or the egg cell. So that brings us to the discussion of genetics. Genetics is the study of genes and heredity. Geneticists are scientists who study genetics. And even prior to the knowledge of genes, chromosomes, and DNA, scientists realized that characteristics were inheritable from parents. And today, depending on the field of genetics, a geneticist does not only study genes, but characteristics that become associated with genes. The passage of traits from one generation to the next is known as inheritance. Characteristics such as skin color, eye color, and the like are inheritable features. A trait is a variant of the character, such as brown or blue eyes. Now, we have, as I mentioned, we'll be watching a movie about him, Gregor Mendel, who was I believe, Austrian monk, who is known as the father of genetics. He famously worked on pea plants in the mid-19th century. Now, note well, he did all his work prior to our understanding of chromosomes, genes, or even DNA. He studied observable inheritance patterns of traits. He observed several traits that differed from pea plant to pea plant. traits such as height or flower color were observed. Peas reproduce sexually, but they normally self-pollinate. And so that is really one key feature that allowed Mendel to do his work. That meant that you have a pea plant in the wild, it's going to reproduce with gametes from itself. So yes, you'll have new genetic combinations, but you're not crossing it with another parent. And so for these traits, the implication would be pea color would have the same set of what we call alleles. We'll look at that in a moment. An allele is basically the genetic code for varying traits. So if a pea plant had, say, for instance, purple flowers, we would be sure that both the maternal and the paternal gene for that would be purple. And conversely, if we had a white flower, we'd know that both copies, the maternal and the paternal, are going to be white, since the mother and the father, in the case of the plant, are the same. So Mendel could easily take these plants out of nature, and he was assured that they were not crossbred. And so then he was able to artificially pollinate the plants. So he would take pollen from one plant. For example, he could take the pollen from a purple flowered pea plant and pollinate the white flower pea plant with that pollen, and he would be assured of a cross between two a pure purple and a pure white flowered pea plant. And so he created these crossbred plants that way. And when we say that, we would use the term that they're either crossed or they're cross-pollinated. And then he took meticulous, detailed records, and he would trace the offspring for several generations to see what traits would or would not be expressed. Now, as we said, Gregor Mendel had zero knowledge of DNA, chromosomes, or genes, but he did believe that there was something inherent to the individual plants, to the life that passed on these characteristics. So he called them factors, and he believed that these factors determined the external traits. What he knew as factors, we today call genes. And through his research, Mendel discovered what we now call alleles. As I mentioned, alleles are alternate forms of the same gene that code for a trait. So in the case of pea plants, the gene that determines flower color has two possible alleles, white or purple. In all, Mendel tested for 7 different traits, and you can see that on the slide I have up. And so the traits he tested for was the shape of the seed, round or wrinkled. The seed color was the second trait, green or yellow. The third trait was pod color. Was the pod green or yellow? The fourth trait was pod shape. Was it full or was it flat? Then he tested for plant height. Was the plant tall or was it short? Then he tested for flower position, axial. And axial just means the flowers would occur at where you have different branches branching off. They kind of make a V, and that would be where the flower would form. Or terminal, which would mean the flowers would form at the end of the branches. And then you would have also flower color as the 7th trait, and whether it was purple or white. Now, since peas naturally self-fertilize, Mendel knew that the parent generation would be pure and not have a mix of what he called factors. The parent generation would be called P, or parent generation, And then once he crossed them, the first generation of crossbred plants would be called the F1 or first filial generation. The second generation, which crossed F1 plants, so you'd cross an F1 and an F1, is called the F2 generation or second filial. And each successive generation would be F3, F4, and so on until you went to the nth generation, and that would be known as the Fnth generation. Mendel could observe how these traits were expressed in each generation of crossing. And that leads to the discovery of recessive and dominant alleles. Mendel discovered that one trait always dominated over another. In the example of peas, when he crossed parent generations of purple and parent generation of white flowers, he always got purple flowered plants in the F1 generation. He knew that the purple trait always dominated the white trait, which is interesting. I'm not an expert on agriculture, but every pea plant we've ever had, I think, has had white flowers. So, that indicates we've bred for white flowers, probably. In today's parlance, we would say that the purple flower is the dominant trait and that it is caused by the dominant allele, which is the gene that codes for purple flowers. We'd say the white flower is the recessive trait and is caused by the recessive allele, the gene that codes for white flowers. So we have the following happen. We have the crossed parents in the F1 generation. And so we can look at what the outcome is when we've crossed and we have the F1 generation. We have parents that are pure, and now we have our F1 generation. And if we look at the traits that Mendel tested for, he found that seed shape, round versus wrinkled, always 100% round in the F1 generation. That meant round was dominant. Seed color, when he crossed a green plant with a yellow seeded plant, it was always 100% yellow, meaning yellow was the dominant trait. Which is interesting. You ask why we have green peas. We don't have yellow peas. Yellow would be dominant. We'd expect it to win out. Well, people prefer having green peas. So somewhere along the line, the decision was made to manipulate what we breed and what we pass on. And so we chose to pass on the recessive green allele, and that again shows breeders' choice in terms of these things. Now, again, with pod color, when you crossed a green with a yellow, we'd get 100% green. Pod shape, full crossed with a flat parent, we get 100% full. Now, we do have both kinds of peas. We have snow peas and we have sugar snap peas. So again, when we consider peas being bred for agricultural purposes, there are different choices made by the breeders to create different breeds of peas. Plant height, we'd cross the tall with the short and we'd get 100% tall. Flower position, axial versus terminal, we get 100% axial. And when we crossed purple and white flowers, we got 100% purple. These were all in the first filial generation. So we would know who is dominant and who is recessive, because we would see no recessive, we'd see 100% dominant when we cross pure parents, a pure dominant parent with a pure recessive parent. And note well, your book has a mistake. Green pod color is dominant. It lists yellow. Um, now, we introduced the idea of the genotype. The genotype is the genetic makeup of an individual. So that is your genes. So when we talk about this in terms of I have a dominant gene and a recessive gene, we're talking about genotype. That's one thing. Then we have something called a phenotype. The phenotype is the expression of the genotype. So the genotype is the collection of genes that you have, and the phenotype is what is observable. In the case of the flowers, an organism which has genotypes for purple flowers possesses alleles for purple flowers. The phenotype, however, is how the genetic code is expressed. Namely, the plant has purple flowers. If the plant had alleles for white flowers, that would be its genotype. And the fact that it expresses itself in the plant having white flowers would be its phenotype. The alleles for flower color in Mendel's pea plants are noted as capital W and lowercase w, which is interesting because we're thinking about purple and we use W, which sounds like white. But the capital W allele codes for purple flowers, and the lowercase w allele codes for white flowers. The capital letters denote which allele is dominant, and the lowercase letter denotes the recessive allele. Since the parent generation was pure in Mendel's experiment, the purple flowers were either capital W, the maternal and paternal copies of this gene, being both dominant purple, or they were lowercase w and lowercase w, possessing both a maternal and paternal recessive white allele. In the case of the capital W genotype, the result is a purple flower phenotype. The lowercase w genotype results in a white flower for phenotype. Now, we're getting ahead of ourselves, but if we have a capital W and a lowercase w genotype, then we would have a purple flower phenotype. So, and that is because the capital W allele is the dominant allele, and the capital W is the purple, and it will be expressed over the lowercase w, which is recessive. In the case of capital W, lowercase w, we say that the lowercase w, the recessive w allele, is suppressed. And in all diploid species, when we consider specific alleles, those alleles can either said to be homozygous or heterozygous. It is homozygous if both the maternal and paternal gene possess the same allele. So in the case of our pea plants, if it's capital W, the two dominant purple alleles, that's homozygous. Or if it's lowercase w, the two recessive white alleles, that's homozygous. Remember, homo being from Greek meaning the same. And then you have heterozygous, and that would be the case where you have a capital W and a lowercase w. And so in that case, the maternal and heterozygous case, the maternal and paternal copies of the gene are differing alleles. So we have three possible outcomes, actually. We can have the alleles be homozygous dominant. In the case of the flower, two purple alleles. where both copies of this gene are the dominant allele. We can have the alleles be homozygous recessive. In the case of the flowers, both alleles are white. And so we possess both copies of the recessive allele. And we can then have the third case, which is called the heterozygous condition, where both the dominant and recessive alleles are present. Now, in the case of both the homozygous dominant and the heterozygous condition, the phenotype, again, remember the trait expressed, will be the dominant phenotype. So in the example of flower color, that would be purple. Only in the case of homozygous recessive will the recessive phenotype be expressed. So again, in the case of the peas, you would have both genes, both alleles be the recessive white allele, and then in that case, the expression would be white, and we would say it has the white phenotype or the recessive phenotype. So looking at the case of the peas and flowers, remember purple is dominant, white is recessive. In the parental generation, we know that every plant is either homozygous dominant or homozygous recessive. Thus, a parent in the parent generation would, with the purple phenotype, would be known as capital W, and the plant with the white phenotype would be known as lowercase w. Now, when we've crossed the parental white with the parental purple, note that every offspring in the F1 generation has one white parent and one purple parent. We get 100% of the offspring being heterozygous in the F1 generation. That means they possess an allele for the recessive and an allele for the dominant. So we have a capital W, lowercase w genotype. But every member in the F1 generation is purple flowered. So we would say it's the dominant phenotype because, again, the dominant gene is always expressed. Okay, so we have the concept of gene segregation. That's the process we saw during meiosis from last week, where you have the maternal and paternal copy of the chromosomes, and they are separated when the cell undergoes meiosis to create a haploid cell from the diploid cell. So you have, in essence, two copies of every allele, and these copies are separated during meiosis so that each egg or sperm only has one copy of the allele. In the case of the parental generation, as we said, parents were in the homozygous condition. So they were crossed with the other phenotype. So a purple flower plant that had its WW alleles separated. In this case, the gametes would all have a capital W. And a white flower plant was lowercase w, lowercase w. These alleles are separated as well, and its gametes possess a single W, lowercase w. However, we are crossing white and purple flowered plants. The F1 generation will be 100% heterozygous, as we said, possessing one capital W dominant and one lowercase w recessive. So then when we get to the F2 generation, our F1s become the parent and they undergo gene segregation. Mendel didn't expect to see what happened, but when the F1 generation had offspring and they were left to normally self-pollinate with themselves, we'd have an F1 crossed with an F1. And remember, even though the F1s were purple, they possessed one copy of the white and one copy of the purple flower. So we would have gene segregation happen, and the gamete could either have a dominant allele for purple flower or a recessive allele for a white flower. And what ended up happening was Mendel saw that roughly 25% of the F2 generation ended up being white. And that was a surprise. So what we know is this, that the F1 generation, you have a dominant and recessive allele because they're heterozygous. You have, from each parent, two possible gametes, one that has the dominant and one that has the recessive allele when meiosis happens. So we have a 50-50 chance from each parent of getting either a dominant or a recessive allele, which means that we have the possible combinations of two dominant alleles for purple flowers or we have a combination that has a dominant purple flower and a recessive white flower, or we have a combination with a recessive white flower and a dominant purple flower. Those are both heterozygous. And then we have the possibility of two recessive white flower alleles. So each of the four combinations is equally likely. So there's a 1 in 4 chance of getting each of those combinations. However, 3 of the combinations, the homozygous dominant, which is the double capital W, or either of the two heterozygous will show up as the dominant phenotype. So those 3 combinations will produce a purple flower. Only the combination of the two recessive alleles, the lowercase w, will express the recessive white flower phenotype. So there is a 1 in 4 chance of having a white flowered plant, the heterozygous recessive condition, in the F2 generation. Basically, Mendel proposed all this theory without knowing DNA, gene, chromosome. Early in the 20th century, another professor, Reginald Punnett, came along and devised an easy way to visualize the genetic phenomenon that Mendel had observed. And we call that the Punnett square, and it's named after him. It's a very nice visual representation that shows both how genotypes and phenotypes are passed on from one generation to the next. It can be used also to predict genetic outcomes. In a Punnett square, we represent the two separated possible alleles from one parent and along the top row. And then on the left side, we have a column that has the two separated possible alleles from the other parent in that column. And then we have sort of a chart with 4 corresponding boxes. And each of these boxes represent the 4 possible genotypes. We can then either write in or show in color the resulting phenotype. So here's the case of the F1 and F2 generations for flower color. Mendel also discovered the principle, which we now call independent gene assortment. That means that 2 different traits are not connected, which means if you look at two or more traits, how they are passed on genetically, behave independently of one another. So in the case of peas, you can look at the seeds. Mendel tested two different seed traits: pea shape, round or wrinkled, and pea color, yellow or green. In the case of shape, round was dominant and denoted by capital R, and wrinkled was recessive, denoted by lowercase r. In the case of color, yellow was dominant and was denoted by capital Y, and green was recessive, denoted by lowercase y. So the parent generation was either homozygous yellow round, so you'd have capital R, capital Y, the two dominant alleles in each trait, or homozygous green wrinkled, lowercase r, lowercase y, the two sets of recessive alleles. The result for the first filial generation, F1, of course, was 100% heterozygous genotypes, meaning each plant was capital R, capital Y, lowercase y. Since the traits of the pea shape and color are independent, there are not 4 possible outcomes in the next generation, the F2 generation, but 16. We can see this by writing a Punnett square with 4 columns and 4 rows, each representing the distinct combinations of each of these 2 independent alleles from each parent. So each parent can have gametes that, and remember, this is coming from the F1 generation, so they're heterozygous. Each parent can have gametes that have either capital R, capital Y, so that would be dominant round, dominant yellow, or you could have capital R, lowercase y, dominant round, recessive green, or you could have lowercase r, capital Y, recessive wrinkled, dominant yellow, or you could have lowercase r, recessive wrinkled, lowercase y, recessive green, as possible allele combinations. When we write a Punnett square, we see 16 combinations possible. We can then group the similar genotypes which do occur and the similar phenotypes. Okay, just to talk a little bit about this Punnett square where we have 2 traits we're looking at instead of 1. So as I said, each parent is going to have 4 potential allele combinations. We see that across the top, down the bottom. And note, we could do this for more traits. We could do this for 3 traits. And if you had 3 traits, then in terms of the combinations, we would have 2 to the 3rd, or 8 across the row and 8 down the column, which would yield us 64 different squares, 64 different possible combinations. And we could keep on going, and we won't though, because it becomes a lot of work. But as we look at this, you can see the combinations and we can group them, and I've done that. But you can note that in the top left corner you have the complete homozygous dominant condition for RR and YY. That only occurs 1 in 16 times. And in the bottom right corner, you have the complete homozygous recessive condition for both RR and YY. So those two conditions only occur once. And we've said that um, these alleles behave independently. We'll see that in a moment. But if you were to just think about for a moment the round peas, and you had two heterozygous, and you were just looking at the round trait, there would be a 25% chance, or a 1 in 4 chance, for the wrinkled trait. The recessive trait would be 25% chance, 1 in 4 for wrinkled. And then you would have, if you stopped looking at the round or wrinkled for a moment and you looked at the color, if you had two heterozygous yellow parents, there would be a 1 in 4 or 25% chance of having a green pea. Well, if you have had probability or statistics at all, when you have independent events, the probability is just the probability of each event multiplied. So 1 in 4 times 1 in 4 is 1 in 16, or you could say it otherwise as 25% times 25%. That would give us a 6.25% chance. But the point is that these traits behave independently. That's why we've written the Punnett square this way. We'll see what happens when we group things. And when we group things, we see that we have several different grouping possibilities. We have, as I said, the capital R, capital R, capital Y possibility. That happens 1 out of the 16 times. The next possibility is capital R, capital Y, lowercase y, and that happens if you look at the Punnett square. You count that twice, 2 out of 16 times. Then you have the capital R, lowercase y. You only find that once in the Punnett square. That happens 1 out of 16 times, and so forth. So you have capital R, capital Y. That happens 2 out of 16 times. Now, the complete heterozygous condition for both capital R, capital Y, lowercase y, happens the most. That happens 4 out of 16 times. Then you have the capital R, lowercase y. That happens 2 out of 16 times. You have lowercase r, capital Y. That happens 1 out of 16 times. You have lowercase r, capital Y, lowercase y. That happens 2 out of 16 times. And you have lowercase r, lowercase y. That happens 1 out of 16 times. And we can lowercase y. That happens 1 out of 16 times. And we can look at the corresponding phenotypes. And so I've written them in this chart where I just have counted all the occurrences of the genotype, and I've listed the phenotype. And then we can say, well, how often does each phenotype occur? And if you look at it, you have round yellow occurring 9 out of 16 times, round green occurring 3 out of 16 times, wrinkled yellow occurring 3 out of 16 times, and wrinkled green occurring 1 out of 16 times. So again, I'm saying that these alleles, these traits, are independent of one another. And so if you were just looking at roundedness and you had heterozygous parents, you would have a 25% chance of wrinkled. Well, we can say, well, how many are wrinkled? And we actually have 1 out of 16 are wrinkled green, and 3 out of 16 are wrinkled yellow, which gives us 4 out of 16, which when we reduce the fraction, it's 1 out of 4, or 25%. So the trait wrinkled is independent of greenness for the seed. And we can ask how many green are there. And again, we would see that we have 3 out of the 16 being round green, and 1 out of the 16 being wrinkled green. And so when we add those together, we do indeed get 4 out of 16. And so again, these traits behave independently one another. We can think about them independently, but sometimes when you're doing heredity, it makes sense to look at multiple traits together. And if you go much beyond two traits at a time, you're probably wanting to use a computer to do it. We see that the principles that Mendel, or as we might say, Mendelian principles, established 150 years ago are still found to hold true today. Our understanding has expanded because we can explain what is happening in terms of DNA and in terms of genes. After Christmas, we will look further at this by looking at heredity and inheritance patterns. So, again, you have these worksheets that I'm sending with this lecture that will have you do some Punnett squares. Please work through them, and hopefully this is understandable, and we will discuss it more after break. Have a Merry Christmas, a Happy New Year, and enjoy your break.